All right, well, good afternoon, everybody. I just want to start off by saying that a lot of the data I'm going to show was the result of a remarkable collaboration with Yasmina, so thank you in advance. And I will try to make sure to acknowledge you on every slide, but if I forget, when I say we, I mean her, when this data pops up. All right, so yeah, here's the thing I hate about antibody engineering. It's fun to develop therapeutic molecules. So the data sets are really exciting. Therapeutic applications are obviously very promising. But the thing that has always driven me absolutely bug nuts about developing antibodies is the time it takes to develop an antibody you want, the number of errors that you put up with in the final molecule, and we try to correct through formulation afterwards, and just the iterative nature of the process. So what I'm going to talk about today is the continuing effort in my laboratory to try to short circuit as many of these steps as possible by learning from the past and trying to install up front a new paradigm where we engineer in advance and perform high throughput characterization of larger numbers of molecules generated faster that are better behaved up front to try to avoid many of these steps that I see as unnecessarily annoying and slowing down our ability to produce medicines faster. I'm going to work towards this ambitious goal of a 1-week antibody discovery cycle. I'll tell you where we are right now. We're definitely not there. But I'll talk about the sets of improvements that are substantial compared to where we were 10 years ago with the new technologies that we've been developing. So my history in this field has been largely trying to understand antibody repertoires and then engineer out annoying things from them so that we can do more productive discovery of more molecules that have better properties faster. I used high-throughput sequencing and high-throughput functional analysis of any kind I could get my hands on during this process, and that's resulted in iterative improvements in libraries, starting with the published work in 2009, which is your typical natural library where you take blood from a bunch of people, and it had the typical kind of annoying behaviors where you had a lot of relatively weak binders on inferior scaffolds with lots of mutations. So, and the other big problem was that there were lots of the same clone over and over again in that library. And so by understanding that from the deep sequencing, we embarked on a series of iterative campaigns to build better libraries that we tried to remove as much as possible, given the technologies of synthesis at the time, the kinds of characteristics that we thought were limiting the quality of those libraries and enriching for the properties that we liked. And that helped. So this is showing a set of libraries panned against PCSK9 as a representative antigen. And you can see we started getting more hits and higher affinity hits directly from these libraries. And that was kind of attractive and critical because the weak hits that we were getting back 10 years ago from traditional natural libraries often weren't high enough affinity to be very easy to perform a functional assay with. And that was hugely problematic because you'd get very few hits and you wouldn't even have a mechanism to know if they were functional or not without performing additional engineering. And that's something that was essential for us to correct. So here's where we are, and I'm going to walk through a triad of technology improvements, including Cartera, that have enabled us to basically reduce by 2/3 the amount of engineering time that was required since 10 years ago when I was working on this class of problems. So what I've done here is I've tried to describe the timelines necessary for a discovery of a hit and then optimization of a hit using a couple different technologies. And you can, and people will, argue with me a little bit about a month here and a month there with these different technologies, but I think all of you will agree on the overarching statement of the rough areas where the pain points lie. So with traditional hybridoma, your big pain point was humanization and optimization after original discovery. And then there's obviously going to be a 3 or 4 month immunization campaign before you can begin recovering molecules. On the right, I'm also listing how many hits you get back out. So traditionally, hybridoma, by the way, is a great technology. It's yielded many of the great antibodies that have worked for us, but it takes a long time. And the diversity of hits that comes back sometimes leaves something to be desired. So traditional phage bore the promise of the molecules being human up front, but suffered from the affinity problem I described and some other developability issues, such that you had to spend quite a bit of time engineering those molecules after you recovered them. And they also, the original generations of libraries didn't have that many unique hits in them to work with. Transgenic animals kind of avoid some of the problems of traditional mouse immunization in that they're already human to begin with, but they continue to have a long immunization schedule, so 3 to 4 months of immunizations. And the molecules that come out, even though they're human frameworks, they still may have mutations you need to correct. So there are some choices that I don't agree with in terms of certain frameworks that are present in those libraries that I'll get into. And there's certain types of selection pressures you cannot apply to a mouse, making it difficult to get certain types of cross-reactivity, species cross-reactivity without creating further transgenics. So the tech I'm going to describe today is going to be the Superhuman technology. This is a computationally optimized antibody library. And the remarkable properties of this library are, first, we made some smart choices, or I'll argue that they're good, and you can haggle with me about that, about the choices of the frameworks to make them more developable and the properties of the CDRs. And then we optimize the diversity. So we end up getting an astonishing number of hits enriching from this library, about 5,000 to 9,000 against any target. We actually don't end up screening all of them, but we'll pull up between 100 and 300 leads from a campaign to pull forward and characterize. And I like that because it gets you more epitopes, more functions, more species cross-reactivity, and all the kinds of features that I want. It reduces the risk of running a campaign and getting nothing that you want. But it created a new class of problems for us, and then I was like, how the hell are we going to screen all of these things? And that was the critical piece that we think Cartera really, why I became so enthusiastic about it from the beginning. First off, based on Yasmina's reputation for excellence in this field, and where she's like, this is the next best thing. And second, because it was able to do 384 wells simultaneously and kinetic information. So it gave us a lot of information very quickly. I'll talk a little bit about this further short circuit called 0 day at the end, which is kind of final paradigm shift that we're already engaging in to pre-pan populations of hundreds of antigens so that we already have them in our fridge, so we just jump directly into characterization when we start collaborations. All right, so I'm going to kind of break this up into 3 bits. One part is thoughtful design, so that's developability, trying to engineer, ask what are the properties of good antibodies, and let's make a whole repertoire just of those things. The second is an astronomical diversity. We want a huge library so I can get thousands of hits against every target so that I can always get the kinds of molecules I want. And then instead of getting a weak hit and trying to fix it, I have an infinite supply of hits and I can just do selection to narrow down the pool that I want. And the final bit is I want futuristic screening. I want to do things faster. I want to search more molecules quickly. I want to have I want the problem to be I have so many hits. I need to solve the next problem of how to screen them all quickly. And I'll walk through each of those pieces now. The overarching design that I'll go into a little bit later for Superhuman is that I chose just the best therapeutic scaffolds. I asked, okay, the human repertoire has 3,500 heavy chain, light chain scaffold combinations, and most of them don't make good drugs. So what are the good ones? And let's make an entire repertoire out of those. Second, I don't want mutations in the frameworks. You don't need that for your product. That's just going to have the effect of making it more immunogenic, potentially less stable, and have expression problems. So let's get rid of that up front. And finally, I want non-synthetic diversity. I want human diversity in all the CDRs. I don't want it coming from a mouse. I don't want it coming from a synthetic design where there would be weird combinations that our bodies would have rejected. And that's what I want to combine. And finally, I want to use computational methods to help understand and then ultimately physically build using real blood a library that's radically more diverse than the previous natural library. So that's basically the design principle. I'll go through those details. This is the process. So what we carry out in our lab. Our lab's right across the street. I just walked over here. So basically we do, like many of you, right? We start with antigen QC, make sure things are in working order. We then use a robotics panning instrument. It takes about 2 weeks for us to conduct our panning. So we have a huge leg up compared to the mice where you're still immunizing 3 or 4 months out. We're already done with our process. And because it's such a huge library, we're doing 16-hour washes and picomolar selections during that programmatic robotic panning process. And that takes us into the screening area. And this is the area where the Cartera was hugely advantageous for us. And we're really excited to see how much we can push the package of what's possible here. And that's where we have 3 different assays that I'll go through to perform a rapid screening of all of our hits and a vector that enables us to jump immediately into screening without additional cloning required. After we have a set of hits based on cell binding, affinity, epitope information, species cross-reactivity, many of these features that are available from the Cartera, after that, if we're doing IgG conversion, I don't have a magic trick here yet, and I'm open to anything you guys can tell me about faster ways to do this than what we're currently doing, which is just a lot of annoying cloning. And then final characterization. So these are the bits. For the panning, back in the good old days, I panned a couple times by hand. Obviously, I don't do that much of this. It's mostly my team. But we don't use people anymore to do this. We basically have a robot. It's a 24-plex magnetic bead manipulation device. And what's really attractive about it is it lets us roboticize, automate, and then standardize our panning processes. And we can do very long, like overnight pannings or very complex deselections on this robot. And they can run on 24 samples at once. In practice, we do replicates on everything, and we do a series of branch pannings. But this makes a very reliable result that got rid of a lot of weird data I saw in the beginning when I started deep sequencing panning selections, and I was trying to figure out why there was only like 10% of clones in overlap. And because they were done by different people and subtle behavior at the bench or levels of sobriety or who knows what's causing it, but you end up with different types of molecules emerging. And we see more consistency now with this platform. It also lets us really I can run experiments that would have been really evil for me to do with my team, where I'm like, let's run this thing for 16 hours, and then let's try 5-minute reductions and things like that I can't ask people to do, but I can definitely ask a robot to do. And that enables us to do this iterative, rapid advancement of the quality of technology we're producing. I'm listing on the left that this robot's also enable us to start zero-day panning. So we've pre-panned a whole bunch of immuno-oncology targets, and they're in the fridge, and we do partnerships with them because we already have the hits, which allows us to jump immediately into screening and skip even that 2-week process. And the 1 week of QC is something that's avoidable. And this is that never-ending effort for me to try to narrow it down to basically just go to the shelf, pull out the molecule you want, and radically transform the paradigm of how we discover therapeutics, because they're already discovered. It's just all about screening. Next bit, of course, is the Cartera. So this is Sarah Ives. She's actually in the audience right here. We were super stoked to have the platform in place. Up until it showed up, we were collaborating with Carterra to make this happen. So everyone was stoked. This is me sitting on it's a very large present. And we use it, I can't overemphasize how powerful this is for us, right? It lets us do immediate screening on single-chain FEs in bacterial soup. And I'm going to show you the affinity information we get back and compare that to the IgG-converted purified affinities that we see. So it's a very powerful screen that's causing us to want to retire ELISA entirely and focus on direct kinetics. And the way the assay is set up, it lets us look at multiple plates simultaneously and ask questions of, well, what's the affinity but against human, against cyno, against mouse? What about against an allele in human populations? Make sure I don't make an accidentally racist drug, right? That doesn't bind the allele variant, which is popular in Asian populations. What about if I'm interested in glycosylation-independent epitopes? These are questions that we can ask very quickly by flowing different antigens and learn that very early. So obviously, we also check it on IgG, and I'll show you after our conversion, the comparison of our single-chain FE to our IgG programs from direct hits and also from an affinity maturation campaign. And then it gives us the ability to do epitope binning and a lot of rapid information that can be immediately discovered on the molecules. Because ultimately, like I said, we do have a limitation. When we're converting to IgG, we might get easily 300 unique clones from a screening, but we kind of try to narrow that down to 48 or 100 unique molecules to carry forward. So we pick the highest affinity population of 100 unique antibodies. And then I don't really want to work with more of that because IgG conversion is kind of annoying. But that's already a really good position to be in compared to other technologies that are going to struggle to achieve that level of diversity. So beyond the Cartera, we also have this sexy technology in place to basically do a 96-well plate-based multiplex cell assay. It enables us to look at single-chain FEs directly out of the library and ask, are they hitting cell surface versions of our antigen? And that could be, in this example, it's PD1 that's human, cyno, or mouse. And this is done by basically barcoding the different cell classes and putting them in the same well together. So it's 96-well plate-based and very quick. But you could also use the same assay to ask about, let's say, domain or allele variation or other, anything else that you could put on a cell and you could tag. You could ask that question very quickly. Stickiness assays would be powerful both for the cell-based approach and then also on the Cartera. And then the last bit is that's how we built our company originally. We have a platform for high throughput, easy characterization of large amounts of antibody sequence. And so we deep sequence everything we run on, so all of our panning rounds of all of our programs. And the reason we like to do that, it's easy, but what it lets us do is this unbelievably ultra high throughput specificity assay, where we can look at every clone that's enriching and every panning, and we can check to say, has that ever enriched against any of the other antigens we've ever panned in the history of our programs? We can look across hundreds of other panning enrichment campaigns and selections and just see, is this molecule very specific to the target, or does it have a little bit of a hint of polyreactivity, right? You can think of it kind of like those polyreactivity ELISAs that many of you probably run, except that this is against every antigen we've ever run, and it grows, and it's way deeper than you could, especially we're running on hundreds of thousands of molecules every time we run an enrichment. So this is showing each little dot is a clone, and that's on log scale. And so that's looking at growth hormone receptor, PD1, glucagon, beta-gal, transferrin receptor, human growth hormone. They've been panned in replicates, so you can see most little red dots are occurring in the intersection between replicates. And you can see a dot here and there occurring across other different antigens. Those are things that we declare either bead binders or sticky, and we'd exclude them from further analysis. This also means that when we do partnerships with people, we can make absolutely sure we never accidentally give 2 different people the same molecule. We can kind of check that and deplete it and then make sure we have a protection of providing people confidence in the molecules we deliver to them. Cool, so those are the tech. I'm going to walk through the design a little bit. Before I do, I'm just going to show kind of the examples of the results of this technology. So this is PD1. We thought it was a good demonstration case, and it's going into the paper. It's a nice little, small protein, obviously therapeutically interesting, and historically annoying in some respects to produce certain types of antibodies against. So in our primary naive panning campaign, we got about 5,600 unique clones enriching against it. We identified a distribution of affinities by Cartera, including down to about 7% picomolar binders and a distribution of weaker affinities, but a huge population that were in the high enough affinity range that could be immediately tested on functional assays. And when we did that, without affinity maturation, we find a distribution of agonists and antagonists. So we partnered our agonists with one partner. We're characterizing our antagonists, including some that came directly out of the library that appear more active than our Nevo control. Up on the top, right, you see population. So there's cell binders. These are just checking the boxes. Do they do all the things we expect, right? So in that assay, you say, yes, we find cell binders. And not only, which you'd expect because they're functionally active, right? But not only are they cell binders, but we can do that screen to identify what other groups have struggled to find, which are human, mouse, cyno, triple cross-reactives. And we found 25. So of the huge population of hits, most of them are not triple cross-reactive. Those are hard epitopes. But we found a whole bunch that are. And that's really exciting because it changes the kinds of in vivo studies that become possible. And many of our clients come to us specifically for these kinds of hard epitopes, where they're like, hey, we want our target. We need to do mouse and human. Can you do that for us? And we've always succeeded. Then the final bit is epitope coverage. PD1 is a cute one because it's a pretty tiny protein. But we've bent our hits. We've got Nevo blockers and Pembro blockers and Nevo Pembro double blockers and even double non-blockers. So we're hitting all sorts of different epitopes all over the surface of this relatively small protein. That's kind of what you'd expect if you have 6,500 enriched hits. You're just going to saturate coverage of the surface, which is kind of what I wanted to have in my laboratory because I never wanted to have to go back. I never wanted to go, oh shit, I spent 9 months on this program. It's not doing what I want. Now I have to restart. That's an annoying situation. I don't want to spend my life doing it. So I built technology so I never have to. All right, so let's go through each of the pieces of the design. And we'll spend some stuff at the end focusing, because this is a Cartera meeting, I want to focus on some of the cool data we've popped up, which is some of it's just like, if you're using single chain FE and you want to know what does that look like on the Cartera platform. If you convert to IgG, what's the relationship of the affinities? Those sorts of things. But because I worked on this and I love it, I hope you guys can entertain me or entertain my desire to share with you some of my designs for a few minutes. So I'll spend a bit of time doing that, and then we'll mostly focus on data. So here's what I mean about design, that when I build a library, I don't just want it to be big. I want to figure out what are all the annoying things that are going to bother us later with a molecule and engineer them out from the library in the beginning. So there's a couple example molecules I'm going to pick on a little bit. So this one's galiximab. On top, I'm showing from the Adimab study a series of biochemical or biophysical trait characterizations. So this one really wasn't that bad. It's basically triggering on a couple different liability features that they published in that study. It's a very nice study. If you haven't seen it, I recommend checking it out. But if I look at this molecule, there's a whole bunch of problems with it where I would prefer to have a molecule that doesn't have these as my lead. So the first is it's got, on the heavy chain, IgHV4-B. This is an ADA risk. So it has a risk of producing anti-drug antibodies because IgHV4-B exists on a copy number variant site. So like half of people don't have this V gene. So it's going to, and indeed you see they have a decent set of people that are basically dropping out where they have undetectable titers because they start producing ADA against this drug. On the light chain, it's using IgL1-51, which from the light chain amyloid database, you can basically look at people who develop this light chain aggregation disease, and you can find that IgLV1-51 is overrepresented in that database. So it's a little bit more prone towards aggregating to begin with. This thing doesn't have that bad. It has a little bit of a signal, some aggregation problems. But in general, I don't use frameworks that are aggregation prone in my library. I want to have these things to be stable, to be able to be concentrated, to be able to deliver sub-Q if necessary. And some of the other issues you can see in the alignments, in red, there's a bunch of mutations in the frameworks that I'd want to go correct and remove. That could be partially why the thermostability could be definitely better than it is. And then there's a bunch of liabilities in the CDR. So this is like a free methionine and H3, acid hydrolysis and L2 and L3, and then the deamination site. So these are the kinds of features that you can go fix, or you can just put up with it and try to move it into clinic and take the risk. But if you can, why not just fix them up front and have a library that doesn't have any of these problems to begin with? Another example, so this is lenzilumab. This one had more problematic features. So it had a significant problem with polyreactivity. You can see it's basically triggering almost all of their assays on that front. The heavy chain is pretty germline. It just has mutations in the CDRH1, which looks good, except that it's using IgHV1-3. that I'm going to show you is I wouldn't use either because it's an ADA risk. And then the light chain, it's got some mutations in the frameworks, otherwise it's in good shape. The problem with this molecule is there's our acid hydrolysis sites in H3 and like glycosylation in L3, which maybe they got away with because it's bent enough, it's not triggering. But the big problem, and I think the reason it's so aggregation prone, is it's very positively charged. You'll notice a whole bunch of the mutations are increasing positive charge, and that's one of your biggest risks for having polyreactivity problems. And it's one of the things that ends up getting selected for if you're using certain types of beads, if you're not careful, and in vitro processes. And this looks like it was a humanized molecule, and they probably went through some processes that were encouraging those mutations. So the IgHV1-3, let me talk about that for a second. So the reason I don't like it is that there are two major alleles for IgHV1-3, and all human populations have both alleles. You can see they shift, so allele 1 is found more often in, looks like African populations. Allele 2 is more often encountered in Caucasians. But every population has a homozygote for the other allele. And these two alleles differ at 3 non-synonymous mutation sites in the framework. 2 of them are right next to each other, and one's down a little bit. So I don't really like this one because it's going to be look more human in part of the population than the other part of the population. So I'd rather use a single framework that looks equally human in all people that I'm going to give my drug to, just to reduce the risk that there's going to be a subset of ADA responses. And there's no reason to use 1-3. You can use 1-46 that has the same canonical topology and has a single dominant allele in all human populations. So that said, those are the things I don't like. Here's the steps that we went through to try to get rid of all that for Superhuman 2.0. So basically, I wanted the drugs to have gone through phase 1 without any problems. We can just look that up. So we did. We want to avoid certain frameworks that have immunogenicity problems using the 1,000 Genomes database and some other studies. Avoid V genes associated with aggregation. We want to pick V genes that are going to display well on our platform, and we have ways of selecting them for that. We want to pick V genes that are thermostable. I like thermostability because it produces better molecules that are going to express better, they can concentrate easier, they're less immunogenic because aggregates are more visible to the immune system. And finally, I want some structural diversity. I don't want to just pick one framework because a single framework has a single canonical topology for the CDRs, and that's going to bias the epitopes it likes to go after. I want to make sure I get saturated epitope coverage. And then I think I talked about it already, but I want to avoid certain, like, I would say deeply problematic V genes that exist in natural repertoires. They exist in natural libraries. They exist in a lot of transgenic mice, and I would never build a drug on top of them, like IgHV4-34 or 2-5, 4-B, 6-57, and so forth. I've got a larger list of these, and I'd be happy to share them if you want to check your molecules. This is just an example of that data. So this is the therapeutic programs. You can see stuff that's made into clinic and been tested in a human is heavily biased towards a limited set of V genes. And that's partially for historical reasons because of humanization. And it's partially because the community at large has identified certain V genes as just behaving well as therapeutics. And so certain groups that have been pretty good at putting molecules into clinic have biased themselves towards well-behaving therapeutics. And I mostly agree with their decisions. I'm highlighting in blue the 4 heavy chains and the 4 light chains that I chose for Superhuman 2. I used deep sequencing data and large screening data from published libraries and some unpublished data from our previous work to identify frameworks that make good choices to use for the platform we want to work with. Because I just don't want to use frameworks that might be good on human B cells, but if I'm not doing single cell discovery from immunizing a person, I'm not convinced that that's going to display well, or I know it isn't on the platforms I'm working with. And then, like I said, I used the 1,000 Genomes Project data and some work by Corey and others who were looking at haplotypes of the IgH locus to make sure that I'm picking a single dominant allele that's found in all human populations. So my drug looks equally human in all recipients. And then we mix them all together. So it's 4 heavy chains, 4 light chains. It's a very big library. It's nearly 10 to the 11. And I bias towards 2 framework combinations that I really like. I like 3-23 KV139, and I like HV146 KV139. Those are my 2 favorites. The idea is if I can get a hit, I'd like to be on one of those 2 frameworks. But to cover my butt, I want the other 14 frameworks. So it's like the best combination of 16 out of 3,500 possible frameworks, and then I choose the best of the best. So I put in more into the library so I can bias my hits towards those 2. So that's the set. Couple slides on how to make it super diverse. So I choose these frameworks I like. And then from studies of identical twins and then us just sequencing lots of human repertoires, I got a pretty good handle on the diversity of how much actual unique B cells are in your blood. And that really helped inform, helped solve the weird mystery of why original libraries were not performing very well and had very weak hits. and helped inform how we built Superhuman to solve that problem. So you have about 10 to the 11 B cells floating around in your blood, give or take, given how big you are. But if we sequence them, a lot of them are copies of the same clone over and over again. So in the peripheral blood, you have about 10 to the 7. It's about 10 to 50 million unique B cell receptors is what actually is floating around in your blood. You're lucky. It's like young people, athletes in like their early 20s might have 10 to the 8. Otherwise, it goes down. And it sits around 10 to the 7 up until people in their 80s and 90s. Really, the thing that kills normal libraries is that your memory compartments, your CD27 population, they're 2 orders of magnitude smaller. So it's really only a few 100,000 clones that are circulating, and there's lots of copies of the same clone, and they produce a lot more RNA. And the reason for that is that when you're exposed to antigen, they make lots of copies of themselves. They start producing a lot more antibodies, so they produce more receptor. And then finally, they don't live in the periphery long term. So when you take a sample, you're just getting a little snapshot of some recent immune events. So something the person cut their foot with, or ate a weird burrito. And most of that memory is not present there. It's actually hidden in the secondary lymphoid organs, like the bone marrow and the spleen. So that's been a problem because when we took total peripheral blood, you end up poisoning your diversity with a limited number of largely uninteresting clones from the memory compartment. So our way of dealing with that is to, first off, take a large number of subjects, because the repertoire diversity really isn't that high per person. Then we use sorting techniques to sort out the naive from the memory cells. We capture the H3 diversity from the naive compartment, which is 100 times more diverse than the memory compartment. And then we capture the memory H1, H2, L1, L2, and L3 from those frameworks that we care about. So we use special engineering techniques to pull out those CDRs just from those frameworks, so we're not mixing CDRs across frameworks. We assemble that on 100% germline, so shown in dark blue, frameworks. So it's those chosen frameworks that we like. There's no mutations there. The CDRs come from people, so they've been pre-screened by blood. And then we check our work through deep sequencing. We actually use deep sequencing also when we take our subjects. The first time we built a library like this, we were looking at the hits, and we're like, why the hell are there like 6% of our hits have a cysteine in H1? And it turned out there was one subject who had a super dominant clone that had a cysteine in H1, and that ended up overrepresented in the library. So we ended up going back, we sequence now all of our subjects, and then we check for each framework, for each CDR, we deplete certain subjects that have weird liabilities over-enriched in their dominant clones. And that provides a mechanism for us to produce a fully human library that actually has less liabilities than is found in nature. So that's the technique. Because we're doing the shuffling, it's within framework, but it's still combining sets of CDRs, then you've broken linkage. We created a linkage release from linkage disequilibrium, and that has the mathematical effect of creating a radically increased diversity, so about 2,000 times more diverse than previous natural libraries. And as a consequence of that, we get a lot more hits. So over on the left, I'm comparing the PNAS the typical natural library, a memory library, and a naive library, compared to Superhuman, shown in orange. What I'm showing there on the x-axis is I've ranked the clones by frequency, and I'm basically saying the top 100 clones, how much of the library is that? The top 1,000 most frequent clones, how much of the library? The top 10,000, how much is that? And so you can see that the natural libraries, the top like 20, 30,000 clones represent about 70% of that entire library. So a very limited number of dominant clones took up almost that entire library. And so even though people claimed it was 3.2 e to the 10 unique transformants, the library size was really about 1,000th of that because it was filled up by a limited number of clones that were high frequency. And that's really a function of the repertoire. Both naive and memory have this problem. Naive isn't as bad. But you can see we've largely solved it with Superhuman. So the top 100,000 most frequent clones represent only 2% of that diversity. And as a consequence, on the right, you're seeing how many enriched clones we're getting against a whole bunch of pannings. And you can see about 5,000, or I guess 4,000 in the case of human growth hormone, because it's tiny, up to about 8,500 for human growth hormone receptor. They're getting a huge number of hits emerging as a consequence of having more molecules in the library. And then when you look at the sequences, because of the way it was built, some other issues that I noticed in my early natural libraries is that sometimes you think you got 100 hits, and you actually look at it and you're like, there's actually only 8 hits. It's 8 heavy chains, and each one had a couple different light chains associated with it. So that was the hit driven by the heavy chain. The light chains just weren't getting in the way. Whereas with this design, you're actually getting radically diverse hits. So this is a set of about 500 hits from the full-length sequences that we have from Sanger. And I'm asking how similar is each hit to every other hit. And so you can see a subset of hits that are identical or near identical, shown on the left. And it's a very bimodal distribution where the majority of the hits are quite different from each other. And that's all happening with entirely natural diversity and just in the CDRs. So you're getting a vigorously diverse population. And that's why we're getting such good saturated covers to the surface. And why when I say there's 6,500 hits, I really mean that. I don't mean it's actually like it's some clerical trick where it's actually only differing by one amino acid. It's a vigorous diversity. Cool. So then that takes us to the fun part, the screening. So you have this huge library. Let's apply it. So one of the cool things that the Cartera has done for us is that, I build these libraries, and then deep sequencing was a way for me to look at the sequences, and I can apply selection pressures and see what sort of sequences change. But that was fundamentally very unsatisfying for me. As a library engineer, I want to see what is the effect on the distribution of affinities that emerge, right? That's the question I'm driving towards. I want to look at functional characterization, affinities, thermostability. And up until the Cartera showed up, I was relatively limited on assays that could enable me to do this. So I would have to choose a small representative set of molecules, and you're really subject to a whole bunch of weird, mysterious biases. When I'm trying to read those papers where people reported on that, I'm like, well, what decisions went in between the initial discovery of hits and them choosing a set that they chose to put on the Biacore to give me an answer of what their properties were? Where now, we just bin the whole damn thing, right? Where we do affinity testing immediately. So we go directly from panning campaign, we plate all of our colonies. Right now we pick 12 plates. We're getting robots in to let us do about 5 times that. But with 12 plates, we go and then immediately can do Cartera screening on the PPE. So into that same week where we start by plating, by the end of that week, we're getting kinetics data back in mass. And this is kind of cool because it's given us the ability to not go through a filtration step of a pre-screen by ELISA or some other tech. So we can really ask, what's the distribution of affinities that are emerging? And so I'm showing that for 4 programs here. What you're seeing is that that population of like the 100 nanomolars, those are still present. It's not like the library doesn't have those anymore. That's really a function that exists in sequence space, the probability of a random antibody binding a random target at a given diversity. The difference here is because we have so many hits, then the long tails of that distribution have started reaching much further than previous libraries into higher affinity. So you can see we're getting at least a few picomolar binders back on every campaign we've run. And then a swarm of single-digit nanomolar and others. And there's always, of course, this big background of 100 nanomolar binders. And we're working on various techniques to either apply additional pressure or relieve that pressure, depending on whether we're trying to produce CAR-Ts or high-affinity members or specific cross-reactive molecules. But it's cool in that it's giving us enough data to be able to ask fundamental questions of what's the relationship between sequence diversity, sequence dispersion, and affinity that can emerge. And these are basically equations that we could just write out and be able to make very powerful anticipations of the kinds of libraries we want to build in the future. And on the right, so that's after selection. So we go do these screens, we pull a bunch of hits back. So by the time we're moving forward to do any functional assays, we have swarms of hits that are in the picomolar, single-digit nanomolar, or basically 50 nanomolar or better range. That those are high enough affinity that we can go run functional tests on them pretty quickly. That's exciting. The other cool thing is that if you're going to go run a test, you might as well ask, is it cross-reactive, or how does it bind and interact with the ligand? So this is the growth hormone receptor panning. So we started screening a bunch of hits. We got loads of blockers and loads of non-blockers of growth hormone. But then Yasmina also is like, Jake, I'm finding a kind of a remarkable couple molecules in here that look like they're performing essentially allosteric ejection. So they would go bind the target, and even though they're not directly competing with growth hormone, that when they go bind or when growth hormone binds, it causes a reciprocal ejection of the molecules. That's really sexy because that suddenly you can identify, if you have a huge number of hits, a molecule that doesn't have to wait its turn for the ligand to go away. It can actively quench a signaling process. And that's a new class of biology that becomes possible if you can do high throughput competition screens of this class. And then, of course, we have the ability to do this high throughput characterization. So this is some of our earlier data where we did both on FACS and on Cartera. We were able to identify populations of clones that were triple cross-reactive from human, cyno, and mouse. We found about 25 of those at this point. Not all the highest affinity. Some of them are like 100 nanomolar. But once you can kind of get on the beach with those, if your goal is to get a triple or cross-reactive, you need to start with one that's actually cross-reactive. And then engineering is a much easier problem than trying to discover an epitope in the first place. So those are the populations we have there. And again, the way the Cartera is set up, it lets us look at a large number of clones and flow different variants of the antigen through pretty quickly, which gives you a lot of information very early on in your screening process. Which then enables us to go take those molecules and then carry them forward. So this is just a zoom in of what I showed previously. These are a bunch of the primary hits from the library. And again, you can see that we're getting these nice high affinity or antagonists, and then there's also a population of agonists not shown here against against PD-1. Okay, so then next question we had. So we were doing all of this work directly from periplasmic extract, so PPE from E. coli, and our phage vector is designed not to produce too much of the single chain FE because it can be toxic to the E. coli. So it's a low copy number, low expression. We have an amber stop built in between our P3 phage fusion and the single chain FE. So we can basically induce our clones without having to do any recloning to start expressing the single chain FE. And our question was, is there enough of that material to be able to run kinetic tests? So it turns out, yes, there's enough for ELISA, and yes, there's enough for the FACS. There wasn't really enough for us to run. We had an octet QK, and it just isn't sensitive enough to pick it up. We did some tests on another instrument, like the Forte Bio Octet Red, that it would pick it up, but I really couldn't discriminate between high. It basically very broadly categorized things as high affinity or low. And so we wanted to ask if we could get more resolution and how far we could go on doing this screening directly with single chain FE on the Cartera. And Yasmina was really awesome on helping us optimize our interactions with on our single chain FEs, deliberately avoiding a hist tag so we can work with hist antigens. And so we have an antibody that captures against the MYC or the V5. And she helped us optimize our process. So you can see early on, this is looking at a population of clones in a campaign. On the y-axis is the affinity observed by single chain FE. And then on the x-axis is when they converted to IgG. And they're both observed in monovalent. You can see there's a strong correlation, but it also looked like early on when we were testing it, there was a little bit of a kind of a roof and a floor of what we were able to detect on the single chain FEs. And I think my guess was it could be a relationship of the limited affinity of the tag, but I'm not sure. But whatever it is, Yasmina did some magic with us. And we were able to, with the more modern tests, you can see a population where you have a couple clones we have clone that basically lost all affinity when you converted to IgG. And there's another one that lost about 10-fold affinity. Another one inexplicably increased in affinity a little bit. But the vast majority, to me, this is like astonishingly well correlated, that the affinity we observed in PPE tracked extremely well to the affinity in IgG after conversion. And that's, it's really a testament. In my mind, it gave me a lot of confidence. It was part of the reason we got the instrument, is that I was like, well, this is giving us a really powerful predictor of the resulting affinities that we're ultimately having to work with, and we can get that directly, very quickly at scale. So the last bit I'll wrap up with here is to talk a bit about affinity maturation applications and IgG expressions. So far, everything I've showed you was single chain FE characterization directly from PPE, which I love because it can happen very fast. But obviously, you're also interested in looking at the final IgG results. So this is the traditional affinity maturation methods that I was taught 10 years ago, where you have some antibody. Pardon the Lego cartoons. These are complicated topics, so my brain finds it easier if I have cute drawings. But you have a hit you like, and the old school way to do this is you go make a little library after each CDR or a subset of them. Then you kind of search through those different libraries to find affinity improvements on each CDR, and then you combine them. And if you're lucky, the combinations don't wreck each other's effects, and you result in an improved affinity molecule. And I kind of hate this approach because it takes a long time. It introduces a lot of mutations that were never found in nature, so you don't know if, they might introduce a lot of tryptophans because they're attractive or positive charges or things that happen in vitro that the body would have gotten rid of. And it's never going to find constellations of residues that only really emerge synergistically when they're all present at once, because you're really searching individual CDRs. So we use a different technique called Tumbler. And the idea here is you have a clone that you like, and because we have those limited number of frameworks, we basically have those frameworks built in with the affinity maturation of humanity's blood built into H1, H2, L1, L2, L3. And so we just take out our H3. We can change it a bit if we like. And then we pop it in, and that instantly makes about 500 million versions of the molecule, exploring the human affinity maturation space that's already been elected by evolutionary forces in our bloodstreams. And it's super fast. It basically takes 8 days for us to do this with one of our own clones. It takes a little longer if we use clones from other libraries. So we do this, and this is showing a distribution of those hits. We do affinity match. Basically, we run out. It's a one-step affinity maturation, and then conversion to IgG. So you can see this was a partner we worked with that had a clone that didn't come from our library, so it had mutations in the frameworks. It had biochemical liabilities in the CDRs. They wanted to improve the affinity. They wanted at least picomolar. They wanted to improve the thermostability, and they wanted all that stuff to happen really fast. So we ran Tumbler. This is the distribution of affinities that we got back from the Cartera assessment. You can see some picomolar binders emerging up there on the top left. The red clone is the starting position of the molecule. Oh, they wanted a range of affinities because they wanted to test the assumption that higher affinity was actually better for their function. And so we panned it under different levels of stringency. And we got this population of molecules back pretty quickly. It solved all of their engineering problems in one pass. So it was like a 3.5-month process. And that became their lead, so that thing's moving towards clinic right now. And of course, we can convert to IgG, and we get these gorgeous screens back. So not only is it useful for us very early on in being able to do the rapid single-chain FE characterization, but it's also the same instrument we're going to be using for, multi-concentration, high-quality kinetics that we're getting on a large number of molecules simultaneously. So that's their lead up there on the top left. Yeah, so that basically provides kind of the set of tools that we need to be able to carry molecules kind of like from where we start, which is conception of a engineering practice, like towards a given target, all the way to the point where we'd hand it off, which is where someone's going to do functional development, and we typically don't do function with our partners. Okay, so with that, I'll just kind of review where does that leave us. So I talked about going from 9 months down to 3 months. Where are we going as we move forward? So for the first couple steps I mentioned, we're talking about pre-panning hundreds of targets. So we get rid of those few weeks of panning, which is really only 3 weeks at this point, but of the QC and then 2 weeks of panning. But we can eliminate that. We can have the things in the fridge and we deep sequence them so we know the enrichment worked. And they're basically ready to move forward with characterization. And then with respect to characterization, using the assays I described, and now that we have a Cartera in-house, we're going to really start to see how much we can push the package to take a program and try to see like how much screening can we conduct, like basically 12 plates in a week. And if you have multiple people on a project, you could have one person working on FACS while another person's working on the Cartera platform, and just really ask a bunch of powerful questions of that data extremely quickly. Again, like I said, I don't have a good solution yet for the IgG conversion. That's something that I'm open to conversations, and we're actively looking at a variety of technologies. We have ideas for using high throughput sequencing and really ultra high throughput barcoding to identify a bunch of variants very quickly for bulk cloning. But by solving these first 2 steps and reducing that down from half a year to a couple weeks, I think that really gets us kind of in the right direction towards the ultimate goal here, which is that antibody discovery isn't really a meaningful barrier of conception of an idea to animal testing.